Hello and welcome to the GCSE Biology tutorial. This covers topic B1 Cell Biology, Part 1 Cell Structure. We'll be looking at cells, microscopes and cell differentiation. We'll also be looking at a practical of preparing a slide. Starting off with, you need to know what the terms prokaryotic and eukaryotic mean. So eukaryotic are complex cells, animal and plant cells, and the main thing is that they do contain a nucleus, and eukaryotic cells then join to form eukaryotic organisms. Prokaryotic means there isn't a nucleus. There is still DNA, and we'll talk about that later, but the most common example is bacteria, and they're often single-celled, so just made of one cell. You need to know the features of an animal and a plant cell. So remember, both of these are eukaryotic cells. You can see that they have some things in common, and plant cells also have more things as well. The things inside them, we call them organelles. Let's look at the function of the organelles. So both of them have a nucleus that contains the DNA. Don't say it's the brain of the cell, but it does control the cell. It controls the activities of the cell. They both have cytoplasm, which is the liquid where all the reactions happen. They have a cell membrane, which is permeable, which means it lets the substances in and out. They have mitochondria, and that's where respiration happens, so that's where energy is made. And they have tiny ribosomes, and that's where protein is made, or protein synthesis. Plant cells also have other features. Plant cells also have chloroplasts, and they contain a chemical called chlorophyll, which is green, and that traps light for photosynthesis. Plant cells also have a cell wall, which is made of cellulose and provides protection and support to the plant, and they have a vacuole in the middle, and that keeps the shape of the cell and keeps it turgid, because it contains a liquid called cell sap. Going back to prokaryotes like bacteria, they don't have a nucleus. Instead, they have DNA in two parts. They either have it in a plasmid, in a circular loop, and they can have it as chromosomal DNA, just free, inside the cytoplasm. So they can have both, or they might not have the plasmids. They don't have chloroplasts, they don't have mitochondria, but they do have a cell wall, they have a cell membrane, and they have cytoplasm. This chart here summarises the difference between an animal and plant cell, which are eukaryotes, and bacterial cells, which are prokaryotes. Moving on to how you would view cells. A light microscope uses light. You need to know certain features. So the eyepiece lens is the lens that you look down, and that also magnifies the image by 10. So it's not the main magnifier, but it does magnify the image. You've then got the objective lenses. They're the most important part of the microscope because they magnify the image. Try not to say zoom, magnify is a better word to use. And normally there's three because they have three different strengths of magnification. The stage is where you would place a slide, where you're looking at the image, and they're clipped into place. And then you've got the focusing knobs, and they focus the image so it's sharper and clearer. So they don't make it bigger, they make it clearer. You will need to know about how to prepare a slide for magnification. So in terms of this practical, there's two main ones. There's one where you might be looking at, for example, onion skin cells or plant cells. The other is looking at your own cells from your cheek, so looking at animal cells. With both of these practicals, to prepare a slide, you would need to use a glass slide, and you would add a layer of the specimen to the glass slide. So that might be a really thin layer from the onion skin or from rubbing a cotton bud inside your cheek. You will then need to add some dye or stain to the specimen, and this is to give the cells colour. You'll then place a very thin cover slip on top, which is like a sheet of glass, thin glass, and then the slide is ready. So you clip it onto the stage of the microscope. You will then start with the lowest objective lens, the one with the smallest number on it. Normally they say ×4, it might say ×10. Then you will look down the eyepiece and you will focus the image by turning the focusing knob. If you want to then see it at a higher magnification, you then can turn to the next highest objective lens and then repeat the steps to focus. You can't just start with the highest magnification, you have to build up gradually. Electron microscopes are more powerful than optical or light microscopes, and they use electrons instead of light. And they can see things at higher magnification, so you can see really small things like ribosomes or plasmids in bacteria, and inside things, so inside mitochondria, inside chloroplasts. They also allow you to see the image at a higher resolution, which means it's a sharper image. You can see the picture here shows that with an electron microscope, more detail can be seen of the plant cell and of an animal cell. You'll also need to know how to calculate magnification using this calculation here. So if you were in the exam given an image and asked to find the magnification, you would calculate that using the size of the image, so you might need to measure that, divided by the real size of the object, which they would give you in the question, and that would give you the magnification. Or you might need to swap this equation round. You might also be asked to find out the magnification when something's viewed under a microscope, and don't forget about the eyepiece lens. So not only does the objective lens magnify the image, but so does the eyepiece lens. So say, for example, you view an object using the times 40 objective lens, then the actual total magnification is times 400 because you'd also multiply it by 10 because of the eyepiece. Okay, so you pause the tutorial now and try and answer these questions. 5 structures or organelles in both plants and animal cells. So nucleus, ribosomes, mitochondria, cell membrane, cytoplasm. What 3 organelles do plant cells have that animal cells don't? Cell wall, chloroplast, permanent vacuole. You can just say vacuole. Where is the genetic material found in animal cells? Well, in the nucleus. How about in bacteria? In the plasmid, those circular loops, or chromosomal DNA. What's the role of the objective lenses? They magnify the image, they make it bigger. Now, not all cells stay the same. They differentiate, which means they become different and they take on different jobs. And that word is being specialised. So they become specialised. There's 5 main ones you need to know about. So starting with the sperm cell. A sperm cell has got a haploid nucleus, which means it's got only half the number of chromosomes in there because the sperm cell will join with the egg cell, which will have the other half. They have a midpiece which contains lots of mitochondria. Remember, mitochondria produce energy and sperm need energy to swim. They also contain enzymes in the head called acrosomes, and they digest the egg when they get to it, the membrane of the egg. They're streamlined and they have a long tail for swimming. So remember those features, not just that it's got a tail, there's quite a lot of features there. Nerve cells, these are long and branched and they carry electrical signals. They're surrounded by a myelin sheath and that speeds up the impulse along the axon. You can see that's labelled C in the diagram. Muscle cells, these are long and again this is helping them to adapt so they can contract and expand when being used in movement. Muscle cells also need a lot of energy, so they also contain lots of mitochondria. Root hair cells, they have large surface area to absorb water in plants, and they're thin walls, so the cell wall is thin to allow for diffusion. We'll look back at root hair cells later on in another topic, but it's important to note that we're referring to plants. And the same with phloem and xylem, that's also in plants. The xylem is a tube that transports water. Phloem is tubes that transport glucose. And again, we'll talk more about those, but they are adapted for their function, so they are specialised. And they appear again in the plants topic. So, a 6-mark question now. If you pause the tutorial and give yourself 10 minutes to answer this question. Okay, so the question is to compare prokaryotic and eukaryotic organisms. Prokaryotic and eukaryotic cells. So, what have they got that's similar? Well, they both have a cell membrane, they both have ribosomes, and they're plant cells of eukaryotic cells, and bacteria have cell walls. But the things that they have different are that the main difference is prokaryotic cells do not have a nucleus. Instead, they have a plasmid and chromosomal DNA. Prokaryotic cells are also smaller and often just result in single-celled organisms, whereas eukaryotic cells form larger eukaryotic organisms. You can use drawings to help you, but the main thing of this question is to talk about the differences and the similarities. So not just the fact that eukaryotic cells have a nucleus and prokaryotic cells don't, but actually talking about the differences as well and the similarities. In summary, we've looked at cells, microscopes and cell differentiation and covered a practical preparing a slide.